Our job in this lecture is to continue the trying to make sense of how urine is formed by looking at the two processes that will modify the glomerular filtrate. So we'll look at tubular reabsorption and tubular secretion. So blood plasma and glomerular filtrate have very similar composition. But if you take it, so if you look at sodium, 142 milliequivalents, 142. Plasma and glomerular filtrate are exactly the same. Chloride ion, exactly the same. Phosphate ion, exactly the same. And glucose, exactly the same. Creatinine, uric acid. But when you look at urine, it is very different. So urine has less sodium in it. It has more chloride ion in it than blood plasma or glomerular filtrate does. It has less bicarbonate ion. It has way more urea and uric acid than the filtrate or the blood plasma does. And again, we're looking at concentration, so this is per volume. Creatinine, huge increase, and notice glucose. If you're healthy, you're going to have no glucose at all in your urine. So there's a big difference between the filtrate that is made there at the glomerulus and the actual urine that we pee out. It is way more concentrated in stuff that we see as waste. It is way less concentrated in stuff that the body would like to maintain in order to maintain efficient body function. And that's all because of tubular reabsorption and tubular secretion. This is how we modify the glomerular filtrate. So let's take a look first at tubular reabsorption. And we've got this schematic here showing us all the different regions of the nephron tube. The glomerulus, of course, the proximal convoluted tubule, the nephron loop, the distal convoluted tubule, and then here the collecting duct. So tubular reabsorption, recall, it pulls materials from the tubules and puts it back into the blood, by way of the fluids that are around the nephron loop. So we're drawing stuff out into the fluid and then putting it into the blood. And so there's the capillaries that surround the nephron, and that's where we're going to be eventually dumping all this stuff back into it. So we reabsorb that material across the membranes of the cells that make up the renal tubule and enter the interstitial fluid that's found all around the tubule. So we move everything out into the interstitial fluid and then from there into the blood. And there are three ways that we can move this material. The first is if the material can pass right through lipids. If it dissolves in lipids, then it will just move down the concentration gradient by simple diffusion. But that's only if the material can dissolve in lipids. The second way is that we use facilitated diffusion. So again, there's that term diffusion here. So we know it's going down the concentration gradient, but with facilitated diffusion, we move it with the assistance of a carrier molecule, of a protein that's found in the cell walls of the cells, not cell walls, the cell membranes, there we are, of the cells that make up the tube, the nephron tube. And so those proteins will pick up from the side that has high concentration and move it to the side that has low concentration. And then if we have a carrier molecule that works with ATP, then we can move things against the concentration gradients, and that's called active transport. So we got three means of moving materials to do tubular reabsorption: diffusion, facilitated transport, and active transport. So whatever materials we reabsorb then are moved into the peritubular capillaries and then back into the full circulation. And this movement is facilitated in a couple of ways. First off, we've got the blood, if you recall, the blood that comes in through the afferent arteria it's going to have high blood pressure because of the backup here in the glomerulus. But the blood that leaves the glomerulus through the efferent arteriole is going to have relatively low blood pressure. And so this low blood pressure makes it easier to draw things back into these capillary networks. Second, there's going to be a high concentration of dissolved proteins. So because we've squeezed out all the fluid, we've turned the blood that leaves into high osmotic or high osmolarity material. It has it has a real strong call. It has high osmotic pressure, I guess that's what I'm trying to say. It has a real strong call to draw in water, and frequently, as we pull in water into the blood, it brings along with it dissolved materials. Because remember, those capillaries, they're leaky, and so as we pull in, thanks to the osmotic pressure, that the colloid osmotic pressure given by the dissolved proteins, that pulls in water and anything else that's dissolved in the water. And then finally, we facilitate all this movement because the capillaries here are especially permeable. They have extra large holes that allow lots of fluid to move into the capillaries, bringing materials with it. So three characteristics of the blood flow here in the peritubular capillaries really helps to facilitate grabbing stuff up and dragging it back into circulation in the blood. So the bulk of tubular reabsorption is done at the proximal convoluted tubule. And in fact, the proximal convoluted tubule is even lined, the cells there, their apical surfaces, have microvilli, sometimes talked about as a brush border because it looks like little furry, toothbrush or something. So these microvilli increase the surface area and therefore increase the ability of reabsorption to occur as well. And so we reabsorb glucose, amino acids, and any of the proteins that have escaped the blood. We reabsorb creatine and many of the metabolic acids like citric acid, uric acid, there should be a comma there, lactic acid, and ascorbic acid. So these these four metabolic acids are pulled back in as well. And the only time that we find these materials like glucose or proteins and amino acids or the metabolic acids in the urine is if they are found in such great abundance that they overwhelm the capacity of the carrier molecules found here at the proximal convoluted tubule. If we overwhelm their ability to drag all this stuff out of the developing urine. We talk about the maximum amount of glucose or amino acids or whatever the material is, that maximum amount that we can bring back into the body as the developing urine is flowing through the proximal convoluted tubule as the renal plasma threshold. So if we exceed that amount, if we exceed the maximum ability of the body to reabsorb, then we have exceeded renal plasma threshold. And this is what happens when folks who are diabetic wind up, if they're having a high blood glucose moment, then they can wind up with glucose in their urine because they squeeze out so much fluid with so much glucose in it that even though all the carrier molecules here in the proximal convoluted tubule are working as hard as they can, there's still too much glucose passing by. If you know the I Love Lucy episode where Lucy and her buddy, I forget Ethel, are working at the candy factory, and they're supposed to pack the candy into the boxes, and pretty soon the candy's coming down the assembly line too fast for them to be able to actually collect it all and get them put into the boxes in time. And they start shoving it into their bras and into their mouths and everywhere they can, just trying to keep the candy from getting past them. That's exactly what happens to all these carrier molecules. There's so much glucose or so much protein. there are so much amino acid that we overwhelm the ability of the proximal convoluted tubule to grab all that stuff back. So if you have never seen that, it's a hysterical bit. So I would encourage you to go to YouTube. All you'd have to do is Google Lucille Ball and Candy Factory, and I'm sure you will find that bit. Oh, it is very funny. So sodium ions are reabsorbed by sodium pumps. So these are ATP requiring transport molecules. And as we bring sodium out here at the proximal convoluted tubule, because we're moving these positive ions, well, the negative ions have a tendency to like hop along for the ride. And so many negatively charged ions, like chloride ion or the bicarbonate ion, will also be reabsorbed. As we move sodium and other electrolytes out of the proximal convoluted tubule, well, guess what? Water will, by osmosis, move across as well. So we reclaim a lot of water here at the proximal convoluted tubule, so that by the time we reach the end of the proximal convoluted tubule, the developing urine is going to be isotonic to the fluid that's around it in the interstitial spaces, so that tissue fluid. So it'll be isotonic from this side to this side of the tubule wall. So almost all the sodium and the water found in the glomerular filtrate that we produce is reabsorbed before we excrete it as urine. Remember, 99% of the water is reabsorbed. We'll also see that there's water reabsorption at the distal convoluted tubule and at the nephron loop. Okay, so tubular reabsorption. Diuresis is an increase in the amount of urine that is produced. So diuresis is an increase in urine volume. Sorry, I had to let the cat in there for a second. Anything that leads to increased dissolved materials in the urine, because it's going to increase the osmotic pressure of the dissolved urine. So let's say we have the urine is moving down the tube, and we got a bunch of dissolved glucose in it, in the water, in the fluids in the nephron tubule. Well, that's going to cause osmotic diuresis, because osmotic pressure is going to call water back in. So we've got, if we're moving out all these electrolytes, sodium, chloride ions, for instance, water will also follow. But if we have lots of glucose dissolved in the developing urine that we weren't able to reclaim, or if there are other ions that are in the water that we aren't able to reabsorb, then that will reduce the amount of water that gets reabsorbed by the body, because the osmotic pressure caused by all this dissolved stuff is going to call fluid back into the developing urine. And so that's the reason why people who are diabetic and are losing a lot of glucose in their urine, they are going to have higher volume of urine output, because all of that glucose prevents the loss of water from the urine by osmosis. Okay, so the distal or the proximal convoluted tubule is one place where we see lots of reabsorption, but it's not the only place. And we're going to look in a minute at some other locations where there's lots of reabsorption in the next lecture. But we're going to go on and look at the process of tubular secretion, so that just so that we have an idea of how this is working as well. So tubular secretion, again, this is the movement of materials from the blood into interstitial fluids and on into the developing urine. And so we can move stuff out of capillaries and out of the fluid that's around the nephron and into the developing urine. So what are some of the things that we secrete? Well, we secrete pharmaceutical stuff, so penicillin, phenylbutyrate, phenobarbital, and other pharmaceuticals will wind up in the urine, as well as a lot of metabolic waste, things like creatinine, ammonia, histamine, uric acid, urea. All these things are going to be can be secreted into the developing urine. Hydrogen ion is very actively secreted, and we have to do active transport in order to do this. Recall that because they have ionic charges, ions are not able to just diffuse across cell membranes, and so active transport is required. But by actively secreting hydrogen ions, we can help to regulate pH of the body. So the body has a tendency to go acidic. We'll see that next chapter as we look at acid-base balancing. And so the ability to be able to do tubular secretion of these hydrogen ions is very important. Last of all, potassium is frequently secreted, especially under the influence of aldosterone. So, especially with aldosterone. So, with aldosterone, we will reabsorb sodium, but actively secrete potassium, so that there's less sodium in the urine and more potassium. Lots of tubular secretion goes on at the distal convoluted tubule and at the collecting duct, but that's not the only place that tubular secretion occurs. And there's a great table in the book that really summarizes what goes on at each zone of the nephron tubule, so be sure to look for that table. All right, I'll see you in the next chapter, and you know what to do if you have any questions.